Welcome back. Today we will be learning about immunoprecipitation, which is a technique used by researchers to isolate and study specific proteins from complex mixtures. We will not only cover the basics but also explore a real world research example to see immunoprecipitation in action. Immunoprecipitation is a technique that allows scientists to isolate a specific protein from a complex mixture using a specific antibody. By targeting and binding to a unique part of the protein, antibodies help pull the protein out of the mixture, enabling detailed study. Immunoprecipitation is used widely in protein interaction studies and antigen purification, making it an ideal or essential tool in the molecular biology and biochemistry. There are several types of immunoprecipitation, each with its unique application. Classical immunoprecipitation isolates individual protein from a mixture. Co-immunoprecipitation or co-IP is used to study protein-protein interaction, helping to identify protein complexes. Chromatin immunoprecipitation or CHEAP focuses on protein DNA interactions, making it invaluable for studying transcription factors and epigenetic modifications. Here's how the immunoprecipitation works. First, prepare the sample by lysing the cells to release the proteins. A primary antibody is added that specifically binds to the protein of interest. After incubation, protein A or G beads are added, which binds to the antibody protein complexes. Wash away non-specific bindings to ensure that only the target protein remains. Finally, elution is done to elute the target protein, releasing it from the beads for further analysis, such as Western blotting or mass spectrometry. To ensure success, address common challenges such as non-specific binding by using blocking agents and optimizing the washing steps. Choose antibodies with high specificity and affinity for the target protein. Ensure reproducibility by following a consistent protocol and including appropriate controls in the experiment. The first step is sample preparation. This involves lysing cells or tissues to release the proteins. Use a buffer that includes protease inhibitors to prevent protein degradation and detergents to solubilize the membrane proteins. Common homogenization methods include sonication and freeze-thaw cycles to ensure efficient lysis. The second step is incubation with the primary antibody. Choose the right antibody for your target protein. Monoclonal antibodies are specific to one epitope, while polyclonal antibodies can recognize multiple epitopes. Incubate under optimal conditions considering temperature and duration to ensure effective binding. Adjust the antibody concentration for the best results. The third step is adding protein A or G beads. These beads bind to the Fc region of the antibodies, capturing the antibody protein complexes. Choose the appropriate beads based on your antibody species and subclass. The fourth step involves washing to remove non-specific bindings. Use stringent washing conditions and select buffers that help reduce background noise. Perform multiple washes to ensure only the specific antibody protein complexes remain bound to the beads. The final step is elution. Use an elution buffer with conditions that release the protein from the beads without denaturing it. Analyze the eluted protein using techniques like Western blotting or mass spectrometry to confirm its identity and study its characteristics. Let's look at a real-world application of immunoprecipitation in cancer research. In this case study, researchers investigated the interaction between the tumor suppressor gene p53 and its regulator MDM2. Using co-immunoprecipitation, they were able to pull down the p53 protein along with any interacting partners, including MDM2. This study provided critical insights into how MDM2 regulates p53, which is pivotal for developing targeted cancer therapy. We analyze the immunoprecipitated proteins using Western blotting. In our Western blot of the input, which represents the cell lysate, we see the presence of proteins A, B, C, and D. However, when we perform the Western blot of the co-IP sample, we observe protein C and any proteins associated with it. In this specific case, we used an antibody that targets protein C, and protein C happens to interact with protein B. Thus, the Western blot of the co-IP will show protein C as it is associated with protein B. This allows us to identify not only the protein of interest but also its interacting partners, providing valuable insights into the protein interactions within the cell. Therefore, from the Western blot, we can conclude that P53 and MDM2 happens to interact with each other.